we have more and more people that are saved from chronic bacterial infections thanks to just one or two or a couple of phage treatments. So we have proof that scientifically it's working.
Antibiotics are one of the foundations of modern medicine. They allow us to treat infections that were once fatal and make many medical procedures possible, from routine surgery to chemotherapy and organ transplantation. But this foundation is under increasing pressure. Antimicrobial resistance already causes more than 1.2 million deaths every year, and the number continues to rise. In response, scientists around the world are exploring new strategies to combat resistant bacteria. Today, I'm delighted to welcome Adele James, microbiologist and co-founder of phages, a biotechnology company developing phage-based therapies. Adele, welcome to the Global Network for Science and Diplomacy podcast.
Yeah, thanks. Thank you for having me.
Of course. So let's dive in. How would you characterize the current stage of the antimicrobial resistance crisis?
So I would say it's more than a crisis. Actually, it's one of the major threats for humanity, and it's, for example, expected to be the first cause of human death by 2050, surpassing cancer. So there's an urge to find solution to this, as you said, crisis.
What key knowledge gaps still need to be addressed to better understand the dynamics of this resistance?
So I would say it's a never-ending process. The first one would be probably horizontal gene transfer dynamics, because we know that resistant genes spread between bacteria through plasmids, transposon, integrants. But the precise rules governing this transfer in real ecosystems and not in the lab are still not clearly understood. And there are many resistant genes that haven't been characterized yet. The second gap, I would say, is mostly related to the ecology of those bacteria, and we are supposed to have a fitness cost, meaning that when a bacteria is becoming resistant to antibiotic, it's supposed to cause a fitness cost to it, meaning that the bacteria will survive a bit less well. Still, there are some moments, some events where those bacteria are still surviving so well and even better while carrying a ton of genes, even though the pressure, the pressure is not there anymore. But I think we can find hundreds of topics on this one. Maybe the last one that I think is very important as well is there is something that is still maybe understudied, which is tolerance, which is not resistant, but still we have some bacteria that seem to tolerate some level of toxicity, and also the impact of biofilms in resistance is quite huge and still understood in my opinion.
Well, thank you. That is a very interesting insight. And as antimicrobial resistance reshapes infectious disease treatment, also researchers are exploring new approaches, and including bacteriophage, as phages is doing. Could you explain what actually bacteriophages are and how do they work as antibacterial agents for those who don't understand?
So bacteriophage, that we call phage for short, is a virus that exclusively infects bacteria. Their name means bacterial eater, but actually they don't eat them, they make them explode.
So are phages just another antimicrobial or a fundamentally different approach to treating bacterial infection?
It's a good question, and I think of course, I'll answer that phages are fundamentally different because antibiotics are static chemical molecule that will kill everything in an environment, whether you have a pathogenic bacteria or commensal bacteria. It has no selection in its spectrum. But phages, on the opposite, are living and evolving entities. They co-evolve with their targets, being the bacteria, and their specificity is very high, meaning that a given phage can only target a very small subset from a single bacterial species, and sometimes even just a strain from a bacterial species. And then we have no impact on the microbiome around the target bacteria of the phage. And we can also play with this evolving capacities of the phages when we have resistance coming, for example. Yeah, it's an arm race, and we can make advantage of it.
So you mentioned that they are the most abundant entities in the biosphere. So what can phage bacteria interactions and their co-evolution teach us about microbial ecosystems?
Yeah, so phages and bacteria have been evolving for more than 3 billion years, and this relationship is probably one of the oldest in the history of life, because phages don't have any other choice, actually. And we have a predatory-prey dynamics at the microscopic scale. And phage regulate bacterial population in oceans, soils, even in your intestines. And without them, certain bacteria would proliferate uncontrollably. And as an example, every 48 hours, 50% of the marine bacteria are killed by bacteriophages. I think this number is huge, and I don't know if we can imagine, but it's huge. And also, I think with this coevolution, instead of seeking to definitively eradicate pathogenic bacteria, which is quite complicated because bacteria will always evolve and become resistant, we can think about controlling them using phages, the way the ecosystem have naturally done for billions of years already.
Phages integrate microbiology with large-scale biological datasets and also artificial intelligence. How computational approaches are transforming the discovery and selection of therapeutic phages?
With AI, we can change things and make things go way faster. For example, we have those machine learning algorithms that can analyze genomic sequences to predict which phages are capable of infecting a given bacteria and estimate their killing efficacy. And then you don't have to test anything manually. You just test the best predicted phages, and you decrease the amount of work at the bench radically.
Do you see AI fundamentally reshaping antimicrobial drug discovery in the coming years?
Yeah, and I think that's probably not even a question anymore. It is already happening, and we have some example, like we have AlphaFold that have revolutionized the protein structure prediction, and that's opening new ways of doing drug discovery. And now we have the generative algorithm that can propose new antimicrobial molecules with novel mechanisms of action as well. What we can get the most out of those AI tools is speed, meaning that we can do everything in accelerated mode compared to what we are doing today. And I think this is where the gain is mostly.
I like to shift a bit to your own path. What first got you interested in antimicrobial resistance in microbiology? Was there a specific question or moment that shaped this interest?
I think especially for antimicrobial resistance, it came late, but so I started my studies in biology to do neuroscience. I wanted to work on autism, then I didn't really like what I was doing, so I went to behavioral ecology. And during this master in behavioral ecology, I read an article on microbial ecology, and it was on coral reefs and direct interaction with their microbial communities and pathogenic bacteria. And I think this is where I fell in love with microbial ecology. And then I did my PhD on this topic in marine microbial ecology, discovered the bacteriophages as main inhabitant of those communities, and then later phage therapy, and then the important and the threat that is AMR.
Your journey moves beyond research into entrepreneurship. What was the turning point that convinced you that advancing phage therapy will require building a company rather than just staying in academia?
Everyone is different, but for me, I was a bit frustrated by the speed and the lack of means and the lack of bringing a real project to the end and like doing something really concrete and with a bigger impact. And for me, going to entrepreneurship was the way to have this impact while still doing kind of some research.
In your view, what were the biggest challenges in building phages in its early stages? Were they primarily scientific, technological, or regulatory?
Of course, we had big regulatory hurdles, and this is what our investors were saying at the beginning, that regulation is a nightmare and that we will never do anything about it. But in the end, we find some openings, so we are happy with that. And then I think, well, phage therapy is kind of an old science. It's been discovered more than 100 years ago. So I think scientifically we knew that there was a huge potential and that something was missing. And for us, it was the AI side facilitating everything and also increasing the precision and making phage therapy faster, customized, more efficient. This is the key that was missing before, and this is why we chose that. So yeah, I would say mostly regulatory and kind of trying to take many pieces in many areas of research, putting them together to be sure that phage therapy would benefit from them and go to another stage. I mean, there are some companies doing phage therapy as well. So we just had a look at what was done and what we could do differently, still keeping in mind what is the biology of bacteriophages to be sure that we understand them well, and then try to, fill the gap with some available technologies, available science that were missing to the field, in my opinion.
Phage therapy is now attracting growing interest worldwide. In your experience, where does phage therapy stand today and what still needs to happen before it becomes part of mainstream medicine?
Indeed, it was developed in the early 1920s and then abandoned when antibiotics were discovered, at least in the West, and still continue to be developed in the east part of Europe, in Georgia, in Poland, in Russia, for example. And today, because of this AMR threat, we need solution to the inefficacy of those antibiotics. And this is why we have a rebirth of phage therapy. And I think we have growing evidence that it will work, that it's working. We have field trials data. We have many compassionate use cases that are being recorded by many hospitals worldwide. So the proofs are here. We just need to make it more accessible. And this is what we are working on. And at Phagos, we focus on animal farming first because this is where we think we can have the biggest impact, as most of the antibiotics are used in animal farming, and also there is a huge pain with infection that can't be cured without any other treatments.
Antimicrobial resistance comes from animals. It's impressive also at Phagos that you are using the datasets, correct?
Yeah, so everything is interconnected indeed, and if we reduce the pressure on animals, we also expect to kind of reduce AMR globally. Also because the bacteria that infect animals are the same genus or even sometimes same species that are infecting humans. So we have a very strong interconnection between what's happening in animals and what's happening in humans.
So antimicrobial resistance is not only a scientific problem, it is also a global policy challenge. How important are international collaboration and policy frameworks in accelerating progresses against antimicrobial resistance?
Indeed, it's a global problem, and even more in developing countries, as it is estimated that AMR will be more problematic in those countries, and because of circulation of people, of goods, we can have infection happening wherever you want and still being isolated in your Parisian hospital, for example. And this is a global concern, a very big issue that we need to address at an international level to be sure that everyone is concerned by this problem, and that, we find solution globally and not only country by country, department by department.
Some experts argue that the biggest barriers to tackling AMR are not only scientific, but also systemic, meaning linked to economies, incentives, regulatory frameworks, and healthcare adoption. Are the main obstacles scientific or structural?
I think we have proof that on the scientific side, it's working. And again, We have more and more people that are saved from chronic bacterial infections thanks to just one or two or a couple of phage treatments. So we have proof that scientifically it's working, but indeed we need to align our ambitions on fighting AMR. Otherwise, yeah, if for example we have France trying to develop phage therapy, but then we have all our neighbors that don't do anything, the impact will be very, very low. So it needs to be global, broad scale, to be sure that we address it properly.
And I think it also needs from the community, the people needs to know that there are other ways to treat also antimicrobial resistance. So it not only stays within the scientific community.
Yeah, we need to educate people. Of course, for me, I'm mostly speaking with people that are scientists in phage biology. So of course, we know what we should do. But indeed, first we need to go to hospital, then and the medicine side, and then to the larger public. More and more people are aware that they should not use antibiotics as they drink water. I'm always quite optimistic, and if we make it obvious and if we find a solution against AMR through phage therapy, I think we can do something about it, and I think it's going to work.
Let's turn now to the future of antimicrobial therapies. If phage-based therapies reach large-scale clinical use, where do you expect their earliest and most significant impact to occur?
With the status on regulation and also the usage of antibiotics, for example, again, I think it's going to be animal farming because we have regulatory openings. We have some companies, including us, working on this field. And I think as well, we are working with thousands of animals at the same time, while with humans, we are working one by one humans today. So I think the scale is very different. That's why I believe in animal farming first.
Stepping back for a moment, antibiotics have shaped our understanding of bacterial infection for nearly a century, as you have mentioned in the previous statements. Do you think phage therapy will shift the way we approach bacterial infections by moving broad spectrum treatment toward more targeted or precision-based strategies?
Actually, yeah, that's exactly the premise, and I believe it's not a premise, this is not fantasy. Because what we do today with antibiotics or large spectrum solution is that we will destroy everything, and it's bad for the environment, it's bad for what I know the best for animals. Then you destroy the whole flora, and then if there is a new infection, there is no commensal bacteria to fight against the new colonizer. So it's just a void to fill with whatever, and the pathogen will win. That's why we shouldn't use this kind of antimicrobials, and we should go with very targeted, and this is the nature of bacteriophages. We should rely on what's available in the environment and just mimic it. And what we are doing at Phagos is treating evolving infection with bacteria, with evolving medicines that are phage-based, and yeah, we develop very customized cures to avoid any co-problems that can be associated with large-spectrum antibacterials.
Looking at the global effort to address antimicrobial resistance, what do you think the scientific community might still be underestimating?
The role of the environment is probably not that taken into consideration when we develop those new cures. We have lab evidence that it's working, but when we go to the field and to the real environment, we have many parameters that are not taken into consideration.
And shifting to the young scientific community, many listeners of this podcast are early career scientists. For those who would like to translate research into real-world innovation, what mindset shift is required to move from generating knowledge to building tangible impact?
A shift to have between an academic mindset and an entrepreneur or a startup mindset, I would say. I think in academic research, we are trained with rigor and we only publish when we know everything is really well studied and we have the answer to quite a bunch of things, and we only conclude on what's been demonstrated, and we do things very well, but also sometimes very slowly. So I think we need to keep that and to build some knowledge at the beginning. I think it's important to know what you're doing and to know not 100% because no one knows, and especially if we're young, we are not those PIs with 50 years experience in one field. But you still should not discover new things when reading a new paper and not being, oh no, I didn't know this whole area of this science. If you're into this science, you still need to have strong basis. And then, I think it's very different, and you'll have to do more quick and dirty work until you're able to have money and have time to go deeper in the precision and in enhancing the solution. But at one point, you need to go quick and dirty, and that's something you won't do in academia for sure.
Let me finish with a broader perspective. If you could change structural elements in the global research or innovation ecosystem, what will most accelerate process against AMR?
Of course, it will be regulation, and if we, especially in generalizing regulation, so that we won't have to go with every countries or, I think it would help, but also that's nice that we are not allowed to do anything. So we still need a level of control in regulation, but I think sometimes it's too much, and especially when you're a young startup and you need to pay for trials that are several millions, it's complicated, and that's kind of a choice to make. So do we go into these clinical trials, or do we choose another way? And then it's complicated to find the right answer. There's something that could be done in the regulatory side. And then related to funding, well, that's not easy, but I mean, it's the game, so I wouldn't change anything here. What's a bit annoying sometimes is when you're not in the trend, it's more complicated to fundraise, but that's something everybody knows in the startup world, so it's something you have to deal with. And with international collaboration, I think it would be an ideal world if everyone was sharing as much as they could. I know it's a very academic way of thinking, and I'm saying that while we are not sharing much, but I'm starting to reconsider this point of view because I think it's not helping as fast as we could in this fight against AMR.
Adele, thank you very much for sharing your insights and for the work you and your team are advancing at Phagos.
Thank you. Bye-bye.
Thank you for listening to the Global Network for Science and Diplomacy podcast.